Kurt talked about empirical models, and we are now going to talk about semi-analytic models. You can also call these semi-empirical, right? Semi-empirical and semi-analytical means they're somewhat analytical and somewhat empirical, and I'll try to point out the parts which are analytic and the parts which are empirical, or I'll see if you can point them out to me. Okay? All right, so just to remind you that the forward model starts with the incident radiance, and you propagate that through the medium using the IOPs and radiative transfer equation, and you've done this using hydrolite. And then the inverse model is where you make approximations to radiative transfer, and then you can begin to estimate inherent optical properties empirically or with these semi-analytic models. And so the approximations to the radiative transfer equation are things like the quasi-single scattering approximation, etc. So our goal is to start with an AOP and to derive the IOPs. Okay. So in 2006, the IOCCG pulled together a group of people who did these type of modeling and had a big, giant analysis of all of the existing models. And they had a large data set that they simulated with hydrolite. And they also collected a lot of in situ data. And they ran everybody's models on all of these data and did comparisons. And what was interesting, but not surprising to me, nor to Jeremy Wardell, who ended up using a lot of this, is that most of the models did pretty well and gave approximately the same answers. Why this wasn't surprising is because they're all pretty similar. And I'll point out some of the differences between them, but the differences are very small compared to the overall underlying natural variability that gives rise to the success of this approach. So this is a nice little report, and you can check out various things in it. Hello. Goodbye. So, but I think this is on the resources page, and so you can have that. Hello. Yeah, you've called a classroom at the Darling Marine Center, so I think you have the wrong number. No idea. I'm actually in the middle of giving a lecture, so it's okay. Just try a different number. Thanks. It was the propane and gas company calling. Oh, okay, anyway. So, sorry, I'm already like the half of my face is so numb. Okay, but on this side, so if the drool comes out, I'm all good. Okay, so anyway, I want to talk about how we measure the reflectance ratio, because you did a lot of these already this last week. So here's an example where you have two cosine collectors, one above the surface, one below the surface, measuring downward E radiance and upward E radiance. And here's an example of some spectra that I measured in a variety of different locations. This is wavelength. This is reflectance, which is the EU over ED. Here's an example of LU over ED. And this is something that you might measure with the TSRB or the HyperPro in buoy mode, where you have E radiance and radiance. This is an example of the upwelling radiance measured in this mode. And what was this mode? How'd you guys do this one? The what? Well, it's got this cone over it.
Well, that's a Lee method.
That's the Lee method, right? Okay, so here's an example of an upward radiance spectrum with and without the cone, removing the surface reflection. Okay, and here's an example, a very old example from the 70s, where this is the reflectance, percent of incident irradiance. This is wavelength. And each of these curves was collected off the East Coast. And so it's basically measuring radiance in a very similar way that the satellite does, but this was done from aircraft. This was the data set that began to justify the ocean color sensor. So it's a very important one, which is why I put it on there. The variations in the shape, of course, are related to the various chlorophyll concentrations that are on here. So from Kurt's lecture, we talked about how you empirically estimate chlorophyll from this reflectance. And this is the Gordon data, again, replotted by Kurt, showing the different channels and for the coastal zone color scanner. And they showed you one of these lines, which was a regression for one of the reflectance ratios. Here's where they began to look at how those different reflectance ratios depended upon chlorophyll. And you can see that some of them, the slope between them varies. And that was the basis of going into looking at the maximum ratio to try to estimate chlorophyll, which is the basis of the Modis and Seewift's algorithm. And so this is, again, what Kurt has shown. This is a variety of simulations. I have this graph from Jeremy showing reflectance spectra at 3 channels versus 555. And so you're looking at the maximum band ratio relative to 555. And so here again is which ratio is maximal versus chlorophyll. And Kurt showed this data showing you the density of points. That's what the coloring on his graph was. How many points had that match between chlorophyll and the ratio? Here what I've shown you is which ratio you're looking at and where that shows up on this graph. So these points are where the maximum ratio is 443 to 555. Okay? This one up here is when 510 to 555 is the maximum ratio. And so does this make sense to you? That all of the bluest ratios are here and the greenest ratios are here? Does that make sense? Why? Open ocean will have bluer waters. The maximum ratio will be a very strong blue radiance relative to green. Those are these very high ratios, and those are the very low chlorophyll values. So as you go to greener and greener waters, this is where you start seeing the switch from one band to another, and you see that there are points that overlay within here, so it's a continuous function, which is encouraging. You're not seeing any steps in the relationship, and that's what we look for when we're doing algorithms. So this is what gave rise to the chlorophyll algorithm. And I've given you the equation here, and the reason I've done this is because we're also going to do this in lab. And so here you have the equation for chlorophyll. The log of chlorophyll is this expansion polynomial, and here are the coefficients, and we're going to be doing the coefficients from OC4. You can pick whichever algorithm you want, but this will be the one that we'll be trying. So anyway, now you have the equation. You can model away based upon reflectance. So in that report in 2006, let's see, Ron Zanefeld and Andrew Barnard and Zhangping Li wrote the first chapter on why are inherent optical properties needed in ocean color remote sensing. And their thought was that if you take the radiance distribution and the spectrum, you can predict chlorophyll through this black box, right? So we laugh, but laugh away, because that's what we're doing here. That's the black box. Chlorophyll is not giving rise to variation in the reflectance ratio. Chlorophyll is a molecule. It's in a cell. Well, it's actually attached to some membranes bound by proteins, then in a cell, which is just one member of the suite of materials that are in the ocean. So in fact, it's really all of these things, the particle size, the index of refraction, properties of the dissolved matter that change the IOPs, that impact radiance, right? And so all of this is what's changing the radiance distribution. And so when we're trying, and this is the same graph, when we're trying to get to chlorophyll, this is actually the number of steps in the box. Radiance, controlled by IOPs. These are the major components in the IOPs. This is whether they're organic or inorganic. Of the organic, which part is dead, which part's phytoplankton, which is other living particles. Phytoplankton have all these accessory pigments, and we're looking at this one little part here that we're deriving. So all of this is in that black box and is leading to that distribution. And so, of course, there's a huge amount of variability in both X and Y in the estimation of chlorophyll. And so this is a really nice paper because it always reminds you about what shows up in the variability of an empirical algorithm. And so what we're going to try to do is break this apart a little bit more and open up this box. And that's the basis of semi-analytic inversion. Okay, how many people have read this paper? How many people have read it more than once? Twice? 3 times? 4 times? 5 times? I have read it at least 10 or 20 times. And every time I read it, I get something else out of it. This is a brilliant paper. And so please read this paper and read it many, many times. And every time you think you understand everything about the paper, read it again. It's really brilliant. So back when we were talking about estimating chlorophyll from reflectance, they were already looking at this relationship between IOPs and reflectance. And they were using it to get chlorophyll, but they were really getting the IOPs. It was just that we didn't know we wanted IOPs. We thought we wanted chlorophyll. But it's brilliant. So we're going to talk about it a little bit because it's really the basis for how all these semi-analytic models started. So they measured EU over ED. Here are their spectra. They had very bad axes labels, so you'll see that I replaced all of their labels because they're small and they actually don't really have anything on them. So this is reflectance, radiance reflectance over wavelength, log scale. The single scattering approximation indicated that this was the relationship between reflectance and the IOPs. This is F over Q for E radiance reflectance, for EU over ED, okay? And they're calling it a constant and not spectral. So the goals of their paper were to explain this level of variability with respect to backscattering and absorption. And then to, by going in the forward direction to model IOPs in order to predict reflectance. And so here we go. That shouldn't be here yet. So scattering could be separated into water and particle contributions, right? We all know that. And therefore, backscattering can be separated into the 2 contributions to backscattering. Well, and what they know from Morell's work was that the spectral dependence of water backscattering is pretty steep. Right? It's a minus 4.3 slope. And that the particle backscattering also is going to have some slope to it. It will have its own coefficient, power. So remember, they couldn't measure things, so they had to be clever. When you can't measure things, you have to be clever. When you measure things, you have to be a good data analyst. So they were clever because they couldn't make a lot of measurements, so they had to base everything on what they could measure. So this is a graph of the spectral slope of scattering from blue to red. And this is the fraction of total backscattering that is due to water. Okay? So when this ratio is 1, that says that the backscattering is due totally to water, that there are no particles. And therefore, we know the spectral slope, because we know the spectral slope of pure water, backscatter, right? And so when this term is small, this term obviously becomes this one, but it says something about the slope. Now, as you increase the fraction of total backscattering that is due to particles heading in this direction, that slope is going to go down. Because the slope of particle scattering is flatter. How flat? Well, we're not sure, because they couldn't measure it. But they suspected it either had a slope to the minus 1, sort of slope like this, or in the case of really large particles, it might be flat. So my question to you is, if you're looking at the total backscattering slope, which has a greater effect? The ratio of particles versus water to total backscattering, or the difference between spectrally flat particle backscattering or spectrally sloped particle backscattering?
The former.
The former, right? As we change the amount of particulate, and going in this direction, we're increasing the amount of particle backscattering relative to water. Our slope changes from this value to this value. But if we change the particle size distribution such that we might have sort of a, this is BB over lambda. This is NP equaling to 0. This might be NP equal -1 in terms of a spectrum. These are big particles, these are small particles. The difference between this only causes a variation in the spectral slope of scattering by this much, maximally, here. So it makes a bigger difference to understand how much of your total backscattering is due to particles versus knowing whether it's a lambda to the -1 or a lambda to the 0 power in your scattering spectrum. Does everyone get that? To first order, to describe the particle backscattering, it makes a bigger difference to know how many particles you have in the water versus knowing what size distribution they have. That's huge. That says that just by knowing particle versus water scattering, you eliminate most of the variability in backscattering uncertainty. Okay? The next order of understanding is what size are they? Okay? Because that's a surprising result. People spend a lot of time worrying about this. They should really worry about this. How many particles do I have in the water relative to water? Okay. So, the first thing they wanted to do was model blue waters. And they said, okay, well, let's make this assumption that in the blue waters, that the reflectance spectrum can be modeled by backscattering due to water plus backscattering due to particles divided by absorption by water. And in fact, they're ignoring the backscattering that's in the denominator. So they're only allowing the backscattering by particles to vary. That's it. And so they generated a bunch of spectra by varying the amount of particle scattering, which gives rise to these various reflectance spectra here. The dotted versus solid is the difference between whether they did spectrally flat or spectrally sloped particle scattering. Now again, this is a log plot for reflectance. So that is their model of pure blue water. So where do you go to find blue water in order to test it? Where do you want to go? Where should we go to find blue water? Not here. Go to the Pacific Gyre. Where else do you want to go? Antarctica. Maybe Antarctica. Crater Lake, Lake Tahoe. There are places on the Earth which have really, really clear waters. They went to Crater Lake in the Sargasso Sea. That's where they happen to have data. There are other places they could go. So each one of these solid curves is a measured spectrum. And the dotted curves are their models here. And the ones that fit the best were models T3 and T4. You can go to the paper and figure out exactly how much particle scattering they had to put in the model in order to match. But this is pretty fabulous. This says that simply varying the amount of particle scattering in your IOPs, adding it to the optical properties of water, and you can model the reflectance spectrum in the clear ocean. Okay? That's pretty fabulous. Also, with the approximations that you had to make in order to, the simplifications to the radiative transfer equation. So this is exciting. So what do? You go from a simple place to a more complex place. And so they moved into green waters. So they have two types of green waters. This is where the case 1, case 2 came from. Case 1, blue waters. Case 2, green waters. It's the original place where they defined case 1 and case 2. And you should go back and read it from that perspective as well. The definitions of case 1 and case 2 have been just bastardized through time. Go back to the original paper and read it. And in fact, what they say back then holds for how we should be thinking about it now. So, the first type of green water that they want to talk about are what they call V-type. Why V-type? Because that's a V. Okay? Waters that have a peak. Yeah, some of them don't have much of a peak, but as you sort of take these waters and increase chlorophyll concentration, you end up with a V-type spectrum. They said, okay, well, to model this type of a spectrum, we obviously have to include chlorophyll, so they added phytoplankton into the denominator. And they said, well, we're going to let phytoplankton and backscattering be proportional to chlorophyll. So they did that. And what they found, was that they would be able to actually model these spectra, and I'll show you the results in a second. But that's all that they did, and this is where they had those big relationships between particle scattering versus chlorophyll and phytoplankton versus chlorophyll, and all of those property plots that Morella plotted for years over global distributions. Came up with a relationship and put it in here. The second type of green water is a U type. Why U? Yeah, because it's a U. Okay? Yeah, this is what I love. I love Weaver's so clever. A plus B equals C, and here's a U reflectance spectrum. So we have this really nice U shape here, again, log-log plot. But the shape of this is fundamentally different from the other one, from the V type, right? These are waters that are dominated by sediments. So here, we actually had to add some absorption by particles. So what do we call this now? What component do we call this? It's not phytoplankton absorption. What is it? It's the NAP. You add these two together, and you get what we call particulate. So you have to go back and make sure that you understand your terms when you read these old papers, because they're different from what we call them now. So this is actually the NAP. And it includes mineral particles, organic particles, et cetera. And they said, okay, well, we're going to let our phytoplankton absorption be proportional to chlorophyll in the same way that we have been. And we're going to allow this particulate or non-algal particulate absorption and particle scattering to not be proportional to chlorophyll. And they model it with some spectral shapes like this, and they have a chlorophyll-specific absorption or a particle-specific absorption coefficient that's based upon actually what the scattering coefficient is. And they're trying to model this. And so what happens? This is their absorption model. This is their scattering model. And the thing is that they know the scattering by water, and they know the backscattering by water, and they measured B. And they said, okay, well, B minus BW is going to give me BP. And this is known. And what I don't really know is the backscattering ratio for particles. That's the only thing that they didn't know in here. Well, what are you going to do? Let's assume it doesn't vary spectrally, that the spectrum of scattering looks like the spectrum of backscattering. And we're just going to sort of muck around until we figure out how to match our modeled and measured spectrum at 500 nanometers, right here. And we're going to call that whatever value we have to make BPB, that's what we're going to do. Just adjust it till we get modeled versus measured there. And then follow the model through here. And so what do they get? Well, they get a good match at 500. The observed spectra from the U and V types are in the solid, and the dotted are the modeled. And so the power of this is that you really needed only chlorophyll, and they actually included some phaophytin in there. So you needed to measure the pigments. You needed to measure B. And from that, you could, and from from this model, you could predict this huge variation in the spectral shape and magnitude of reflectance. So it was really the first IOP model. So how much empiricism was there? Some. There's some in there. There's this relationship here about, chlorophyll to absorption, and there's some, they had to do some tweaking here. Tuning is also some empiricism. They came up with some ratio for, in the V-type model, that scattering was proportional to chlorophyll. But then they allowed the shapes of these to vary analytically. And so it's somewhat empirical and somewhat analytic. And so then what they did is they said, so after going through all of this and getting the IOPs with a model, they try to estimate chlorophyll, which is just crazy to me. But this is the reflectance ratio from blue to green versus chlorophyll for all of the models. And they find this very nice relationship. As you increase the chlorophyll concentration, this reflectance ratio goes down. Not surprising. The waters get greener the more chlorophyll you have. But there's an awful lot of variability here, right? That variability is caused by the variations that have nothing to do with chlorophyll. The variations in absorption, the variations in scattering, the variations in the relationship with those IOPs relative to chlorophyll. And so that's why you get scatter in these relationships, because it's not chlorophyll that's changing this. But in fact, what they had was the model of what is changing this. And so it's a great paper to go back and read. It gives you just a sense of history and how far we really haven't come. We haven't gotten a whole lot better than this. Okay, so then time goes by, and in the 1990s, people started to think about actually inverting reflectance to get the IOPs. And so there was this flurry of papers. It seemed like a huge flurry. There were like 4, but they all came out within months of each other from labs all over the United States who were not working together. It was like there's that magic point in time when everyone wakes up and goes, oh, right? It's exciting. It was so exciting to be at meetings because everybody was presenting this stuff, and it was like, oh my gosh, I did the same thing. Oh, you did that. Oh, so it's great. You'll have these moments in your career. It's really wonderful. So here's how it works. The step 1 is to recognize that the IOPs are additive and that you can separate absorption and scattering into components. And so we went back to what we could measure, which is separating particles from dissolved and then separating the particles into phytoplankton and non-algal particles and coming up with this set of absorption shapes. This is water. It's a relative constant, and at least it's known. And that we could come up with some nice curve for phytoplankton. Non-algal particles has a flatter slope. CDOM has a steeper slope. Particle backscattering you could separate into water and particles. And again, those particles could differ from a flat slope to maybe a -1 slope, depending upon size. Step 2 is recognizing that one of the outcomes of Beer's law is that the component IOPs are really linearly proportional to their concentration. And so what that means is that if you're interested in phytoplankton absorption, or any component, you can define a mass-specific absorption coefficient. So if you took all of the phytoplankton absorption spectra that you measured in the lab on all these different species, or in the field, and you have all these different shapes and all these different, and they all look like this, and some are like this, and some have more accessory pigments, but you took all those absorptions and divided them by their chlorophyll concentration, that everything tends to collapse, and then you can define some average shape, right? And then you can take that average shape, we call that A star chlorophyll, the chlorophyll-specific absorption coefficient. And if you take that shape, that average, and you multiply it by your chlorophyll concentration, you can come up with a pretty good estimate of the phytoplankton absorption spectrum. Because most of the variability in the ocean in the phytoplankton absorption spectrum is due to concentration. Right? If you have a low concentration, you'll have a little tiny phytoplankton absorption. If you have a lot of phytoplankton, you'll have a big absorption concentration. And the variability between species and light history and nutrients is second order and much, much smaller. So biomass is the biggest driver of variability. Composition is a second order problem. Jack is scowling at me.
You're not supposed to see that.
That's okay. No, I want to see that because are you scowling because that doesn't sound correct, or do you have to think about it?
A little bit of both.
Okay, so let me give you this question.
Is the A-star chlorophyll just erased the same as the A-star phytoplankton that you have up here?
Yes. Yes, thank you. The star means chlorophyll specific. Okay. compare that variability. Oops. With that variability. Which is more variable in terms of absorption by phytoplankton between the two? Is this a bigger difference than these two spectra? I hope your answer is yes. So here's a phytoplankton absorption for 1 and 2. Here is a star phytoplankton for, this one must be 2 and this one must be 1. So yes, when you divide by the chlorophyll, you begin to see variations in the shape that have everything to do with pigmentation and packaging and species and light history. But the variability in shape is very small relative to the variability in magnitude. And what this captures is the variability due to magnitude, which in terms of the global variations in ocean color, this is what matters. Not this. This matters if you're interested in phytoplankton functional types. Okay? This is order 1 variability. When you do a really good job with this, then you can begin to worry about order 2. Do not worry about order 2 variability if you're doing a bad job here. Right? Do this first. Okay. So what that means is if you can define some nice spectral shapes that are pretty applicable for your component, and you can define some mass-specific, you can normalize them by mass. In this case, we're doing chlorophyll. Other components will be some other specific. Then you can use that in this model. And so if we think about this component IOP as being concentration times the concentration-specific IOP, that's the same thing as a scalar times a vector, which is the same thing as a magnitude times a spectral shape. Right? This has wavelength, this doesn't. And we can think about this as an eigenvalue and an eigenvector. Those are all the same ways of talking about it. Okay, so that's what we did. We put it all together, and we have backscattering by water and some mass-specific particle scattering coefficient and a mass-specific scattering spectrum. So this is mass, and this is mass-specific scattering spectrum. We have water absorption, which is a constant. And here's your phytoplankton. Eigenvalue, eigenvector. NAP, eigenvalue, eigenvector. CDUM, eigenvalue, eigenvector. And then here's the backscattering part. So B back over A plus B back. So we know these spectra. We have to define these. And you know, you have a pretty good idea what these are, right? What are these two in terms of spectral shape? Exponentials. And you have a pretty good idea of what the range and slopes are, right? And we have some idea about the scattering spectrum for particles. So we can do an analytic function for that, an analytic function for this, an analytic function for this, an analytic function for this, semi-analytic. This is where it comes from, because those are all just math functions. The empirical part is, what's our slope going to be? And what's our phytoplankton absorption spectrum going to be? That's the empirical part. Okay, so now you put in those known eigenvectors, once you define them. You have a measured reflectance spectrum. Yep?
So the way I was thinking about this is empirical versus analytic is that those eigenbases, or that eigenbasis itself is the empirical part.
Yep.
And the rest of it, like the framework.
The framework is the analytic part. The mechanism that transports IOP to AOP is an analytic part. And also the fact that you empirically, we find that the shapes of some of these can be described by an analytic function, and you can go take it back to absorption physics, and you're looking at the tail end of absorption spectrum, which has everything to do with state transitions of absorption, etc. Right. So you can push that line as far as you want between analytic and empirical.
It just has to be useful.
Right. Yeah. So what we do is we measure a spectrum of reflectance. We know, or we've defined, the spectral shapes of these, and we do a multiple nonlinear regression to estimate these coefficients, these scalars, in a least squares minimization. So essentially what we're asking is, how much do I need of each one of these absorbing and backscattering components in this mathematical formula to reconstruct our reflectance spectra? Yeah.
Does that mean that
Yep.
there's not a unique solution for this? Or
Well, that's the question. Is there a unique solution? Okay. So I did this graphically because sometimes it helps for people to see things with pictures. So I did is I gave you a spectrum of reflectance. And I said that equals the spectrum of backscattering by water plus some spectrum of backscattering by particles divided by, this is the divided by line, and here are all of my absorption by my components. There's also the scattering down here too, but I didn't have space to add it in. And this is the solution. These are constant, and these were achieved by minimization. So it takes this much phytoplankton, 0.05, this much CDOM, and this much NAP, and this many particle backscattering to reconstruct the shape of that curve. It's cool, huh? You're going to get to do this today, presumably. Okay. So, one of the things that we want to think about, why am I saying this here? Oh yeah, that I was missing this on the other side. Okay. So, of those original 4 papers that all came out in 3 years, there are differences between them. And I encourage you to read all of the papers. They're old. So you're going to read them and go, oh, why would you do that? They're old papers. But the beauty of it is that they all arrived at the same approach and essentially the same solution independently, which gives you a lot of validation in their utility. But there are differences. And so when you read the papers, you have to think a lot about the definition of their eigenvectors and how they derived their functions for absorption and scattering. The method that they use to invert is very different. Some use a nonlinear least squares, some use a linear matrix inversion, there's some optimized least squares, there's sort of differences in there. And then how they validated, how they did error analysis, etc. And each of them has something really interesting to offer. And so, I mean, I made up a big table for myself and kind of went through each paper and figured out where, and it's a good exercise. So we're going to get to we're giving you code for all of these that you can just take home with you. You're welcome. Just take it home. Use them, change them, whatever. And so again, these are the things I want you to think about when you use them. And some of them were original, the first time Lee came up with his inversion model, and then you learned something, right? And then he went, and that became the basis of QAA. So, we did something in 95, and then we did something different here and something different here. So, we all learn and kind of move on and change. But it's worthwhile going back to the originals before you go to the more recent ones. Okay, so I'm not going to go through each one. I've given you a ton of overheads that actually go through a lot of them. You're welcome to look at those at the end. But I want to show you some examples. So I want to talk about some of the ones that use a nonlinear regression. Yep?
Sorry to keep asking.
No, please ask. That's why we're here.
The last slide. So when you do the sensitivity analyses on this, do the choices of the eigenvectors make that much of a difference?
Why, yes, they can. Under certain situations, yeah. And that's something that's really important.
Yeah.
And none of these looked at uncertainties, right? They would do a bootstrap or whatever. But it's not like some of the other approaches where you can actually calculate an uncertainty in your estimates. Okay, so here are the eigenvectors for particles. And when I did it, I divided mine into large particles and small particles and sort of allowed the inversion to try to tell me which of one made the most sense. How much of each one do I want? These were the basis vectors for absorption that I used. So here's the phytoplankton absorption spectrum that I used in my model. Where did I come up with that? Well, by that point in my career, I'd measured a lot of phytoplankton absorption spectra in the field. And so what did I do? Well, I averaged them all. I normalized them to get their spectral shapes, and then I averaged them. And they were all field spectra. And I just said, you know what? You got to come up with something. So let's look at the average, and then we'll see how bad it is. So would you want to use this if you were in the equatorial Pacific Ocean? Probably not, because this is more of a coastal one. But, you know, I was looking at this problem versus this one. Now we're at this problem over here. Okay. These are data sets that I had collected for estuarine waters. This was Puget Sound. This was a fjord up in called Deba Bay in Puget Sound, which is really interesting optically. This is some coastal waters, the Gulf of Maine and in the Pacific. And here's some very clear oceanic waters. The range in chlorophyll for these samples, 0.07 to 25. So huge range in concentrations, huge change in range in absorption coefficients and backscattering coefficients. So the goal was, with this in tremendous dynamic range in properties, this tremendous range in reflectance spectral shapes and magnitudes, could one model do a good job? That's the question. And I've given you all this data. In the lab, you have all of these spectra. So you run it. And this is what you get. I picked out one from each one of these, trying to get a big range. So there's one to try and capture the largest variability. So what do you see? The black is modeled, the colored is measured. It's not bad. Right? There's some changes, there's some variations here. But look at the tremendous range. I mean, look at these are different scales. This is peaking at a very different place than, say, this one is. So what this says is that if you have a 6-component model for large and small particle backscattering, literally just lambda to the 0, lambda to the -1, some phytoplankton absorption spectrum that doesn't actually represent anything because it's an average of everything. And I actually ended up combining CDOM and NAP into with just a single slope of 0.0145. What makes up most of the variability in reflectance is the ratio of all of these, not necessarily the spectral shape of them. That is tremendous. It's tremendously powerful. Because what it says is, you can put in any spectrum that you want, but most of your uncertainty is, do you know the ratio between these and how much of them are in here? That's what's going to vary. We're going to do this simulation. I'm going to give you this data. You're actually going to do this in Excel. It's tremendously powerful. And so the other thing that it says is, there are some places where the models don't fit. Is it surprising? No. And in fact, if you begin to look at the residuals, there's a tremendous amount of information in the residuals. Another opportunity. Okay. And of course, it doesn't fit here. Why doesn't it fit here? What? Inelastic scattering, fluorescence, right? The model doesn't have fluorescence in it. The measured has fluorescence. But that is a really cool thing too, because then you can take your measured minus your modeled and you get a fluorescence spectrum. And then you can do a lot of characterization of that. And in fact, if you notice here, most people just draw the line right across and estimate the fluorescence line height. Oh no, it's very complicated under there because you have absorption peak right here on top of water. So just drawing a straight line really underestimates what your actual fluorescence is. So this is a great way to get at fluorescence spectrum. So this is just incredibly astoundingly cool. Okay, so how did it work? Well, I estimated chlorophyll by looking at the line height of my retrieved phytoplankton and compared it to what I measured. On a log-log plot, everything looks good, so we like that. Phytoplankton absorption, this was my estimated phytoplankton absorption at 440 versus what I measured on a filter pad with a beta. And we know like how much variability we probably have from beta. But, you know, over a large dynamic range, pretty good. Estimated backscattering coefficient at 440. We didn't have backscattering sensors back then, so I actually had to measure the particle size distribution in a Coulter counter and convert it to a particle cross-sectional area, multiply that by the concentration, and get some sense of what backscattering should be using efficiency factors. Not bad. So you're able to, with that model, to recreate the shape of the reflectance spectrum and validate versus the IOPs. So the third thing, and this is one of the things that when you start talking about what you do with the data once you get to that point. So that first estimate of phytoplankton, remember, had a constant shape. This was my eigenvector. And so the phytoplankton absorption was that times whatever the eigenvalue I retrieved was. But for every one of those spectra, the phytoplankton absorption that was estimated had the same shape because that's what I put in, the one shape, right? But, hello, when I looked here at the model versus measured, I noticed some interesting things. I noticed that if you looked at the model versus measured, it's not random. There's not random noise in the relationship. If you took this measured minus this modeled, you're going to get like a little peak. A little peak centered right here at about 440, which could be excess chlorophyll absorption. That actually was in this water, but that wasn't in the eigenvector. This one had a little peak over here. This one had a little peak here. This one actually had sort of an exponential difference. I think that was just had more CDOM. The CDOM shape was different than what I measured. So when you look at the residuals and you plot the residual spectrum, what pops out to you are either little exponentials or little peaks associated in places where there are pigments, where your original shape has maybe too much absorption of a pigment peak or not enough. So I said, okay, to first order, I'm going to take the residual between modeled and measured, and I'm going to just shove it back in to the phytoplankton component and see what happens. And what happens is you get variations in the shape. And so the black line, let me try to remember this. The thin line is what I actually measured on the filter pad, and the black line is this estimate plus the residual. And so you can see they're very different spectral shapes. So, I mean, this one had really close. This one says, oh, it should have been more peaked. So there's information in your error that you shouldn't throw away. It's another opportunity. Okay. So, sensitivity analysis, you change the shape of this and see the variability in your retrieved eigenvalue. You can change the slope of your CDOM and see how that changes. You can change the slope of backscattering and see how that changes your retrieval. And so a lot of the papers do this type of an analysis, kind of a bootstrap analysis. And it was generally about 30% variability, but that's not a factor of 2, which is exciting. Okay. All right. So I think Kurt went through a reflectance model where we retrieved beam C. And so I think that I will let you go through this one on your own. You can work your way through the math, talk about why it works and what we end up with. And the upshot is that it allows for spectral variability in backscattering. And so it gives you a better fit. And so it turns out that the blue curve matches the black curve better because it allows for spectral variability in backscattering, which when you have a lot of phytoplankton, like in this case, you have a lot of phytoplankton, having a backscattering spectrum that varies with absorption works out. So I'll let you go through that on your own because we have had to start late today. And that was validated. We were able to estimate absorption. We were able to estimate beam attenuation from space. And this is the slope of the beam attenuation. And so we were able to estimate the slope of C, which tells you what? What does the spectral slope of the beam attenuation tell you? Particle size slope. So by estimating the slope of the beam attenuation, that gives you gamma, which is related linearly to the particle size distribution. Would you like to know that from space? Yes, please. Right? And so where we had measurements, which was only in this South Africa situation, it was pretty reasonable. At least we could get from high to low. These points didn't work. These were during these really intense monospecific blooms of phytoplankton where kind of all bets are off. And I don't know whether it was an error in my beam C or the fact that the particle size distribution was so narrow. But it's exciting that that much information is coming from space. And this approach, being able to estimate a slope of either beam attenuation or backscattering, is what allows you to say something about the particle size distribution. And there have been other papers following along from this. Tiho Kostanadov, who came out of Dave Siegel's lab, published some papers looking at the slope of backscattering to try to get at size distribution. And it's really exciting. Okay. And this is the retrieved spectrum of backscattering here. This is one that the model just fit an exponential, but this is the actual spectrum, and it looks an awful lot like an inverse phytoplankton absorption spectrum, which we know to be true. I had 2 measurements at 2 wavelengths, and it seemed to fit the backscattering spectrum that was retrieved from ocean color. So, one of the other approaches that I want to alert you to is something called linear matrix inversion. The other models have all been talking about a nonlinear multiple regression. And so Hogan-Lyon published a paper on linear matrix inversion. They used 3 wavelengths of reflectance, which meant how many unknowns could they have? If you have 3 wavelengths, you can get 3 unknowns. Okay? So my first question is, if you had multiple wavelengths, and this was our original equation, does that look linear? It doesn't look linear, but it turns out, yeah, you just start moving things around, right? So let's move this over here and let's move that over here. And so here we have the denominator. Here's the numerator, and then we moved r over, right? We can all do that. Now let's bring like terms together, okay? So we don't know these. We know these. So let's put all the waters on one side. And so we have F over Q times BB because we've taken this and multiplied it and moved it over here. We're going to take water and move it over here. Take this water and move it over here. So right now we're just sort of moving terms back and forth. And then if you look at this, we have all of our unknowns on this side and all of our knowns on this side, right? So we have phytoplankton here. We have CDOM or CDOM plus NAP and particle backscattering. F over Q, we're going to have to call that a known, 0.33 if it's a radiance, 0.0895 if it's radiance, and then these are known. Okay. So that gives you a linear regression. And this will be a spectrum. These will all be vectors, right? This is a vector. And so now you can do a linear regression. And it says unknowns and knowns. And so the beauty of that is that it yields an exact solution. The other beauty of that is that it's really fast, which made it incredible for image processing at the time, because back in the 90s, we were limited by processing time for each pixel. And once you get into a system where you're over-constrained, where you have more wavelengths of reflectance relative to the number of unknowns you're trying to estimate, you're over-constrained, you can begin to do some computation of uncertainties, and I'll show you how to do that. And so if you look at the PDF and you go down to the end, there's a description of what Hogan-Lyon did with 3 wavelengths and what they were able to estimate, and it's really quite elegant. Emmanuel built on this by taking over-constrained systems. Now that we had satellites that had 7, 8, 9 wavelengths, and we only needed a couple of unknowns, and by using that, we could actually get uncertainties, which is really scary because nothing to this date has had an error bar on it, has it? It all looks so nice when there's no error bars. It's so neat. Why would you want to ruin it with a big error bar? But it's important to be honest. And so, so we can look at our uncertainties, and we can actually include our uncertainties in our measured reflectance, and we can measure our uncertainties in the shapes of our basis vectors. Yeah, James?
How weird is it to have the F over Q as kind of, would you be able to start using the uncertainty analysis for that, or is it still...
Well, and so in fact, you could start to put that an uncertainty in there as well. Yes, exactly. Did you measure F over Q this week? How would you measure F over Q? How would you calculate F over Q? Huh? You could do hydrolyte simulations to figure out what F over Q was, because you know your A's and B backs, right? And you know that R of lambda is equal to F over Q of B back of lambda over A plus B back. And you've input those. You know what those are. You know what that is. And hydrolyte can calculate. I mean, you're basically calculating this. It's basically the shape of the radiance distribution, which is something like the shape of the backscattering. So the problem with hydrolyte is that it puts in an F over Q based upon the volume scattering function choice that you gave it, and the radiance distribution above and below. So, but if you could measure this in the field, and you could measure radiance and upwelling radiance and downward E radiance in the field, you could calculate this and figure out, number one, what its value is, and number two, does it vary spectrally? Does it vary as a function of time of day? Does it vary as a function of the types of particles you have in the water? Yes, yes, and yes. But at least you could understand what your uncertainty is there. Okay. Good. All right, so this is an approach that Emmanuel took. We have all of these eigenvectors. I'm going to call this now BBP, which have some shape. Okay, we're going to say that there's some shape. It's lambda to the eta, and eta can go from -1 to +1. So you might have a shape particle scattering that actually increases with wavelength. Could be flat. We have some variability in phytoplankton spectral shape. We have some variability in detrital matter, C-dominant NAP, that can be flat or steep, and everything in between. And so by running it through the linear regression, you can set up a giant multiple loop program where you run the inversion